Hello students, good morning all of you. Welcome to our next lecture, that is Monocotyledonous stem. That is primary structure of Monocotyledonous stem. In our previous lecture we have discussed about the primary structure of Monocotyledonous root. So today we are going to start about the stem. So in Monocotyledonous stem, in your syllabus the Mace, that is plant, is included. So we are going to see the internal structure of Monocotyledonous maize plant, stem of the maize plant. Okay, so let's start with the structure of Monocotyledonous stem. So it's look like somewhat like that. In this picture you can see the all the internal structure. Basically it is divided into three parts, as you can you know it better now that we have studied the dicotyledonous structures, and in monocot also we have completed the root structure also. So these internal structures, they are differentiated into three parts. The outermost is epidermis, then the middle portion is the cortex region, and the third one is the still region. Okay, so these three basic regions or points, things, they are present in any internal structure. So the difference is between these, the presence of which type of tissues, how the cortex is maybe homogeneous or heterogeneous, vascular bundles or the steel may be different types. These all these things, they are different for different that is organs of the plant body. So let's start with the epidermis. So epidermis, as you know, that it is a single layer of cells with well developed cuticle. So it is a layer which is made up of cells which are compactly arranged. Okay, there is no intercellular spaces in between these cells, and these epidermal, it is the outermost and the protective layer which is present surrounding all organs of the all over the plant body. So epidermis, it is a single layered that is um, protective covering which is made up of cells which are compactly arranged, and over to the, that is these epidermis also secrete some substances, that is a cutin or the wax like substances, and this protective layer is again developed above the epidermis. So it gives the additional protection to the epidermis. So this cuticle layer along with the epidermis, it protects the inner tissues of the plant body. Okay, so the trichomes, they are absent in case of maize plant. The trichomes are absent but stomata are present. The stomata, they are present on the stem, but due to cuticle, there that is the transpiration rate is minimized. So trichomes may be absent, but the stomata, they are present. Because stomata is again responsible for the gaseous section, so that's why it must be present on the epidermis. Now then it protects the inside tissue against evaporation of water. Yes, this epidermal tissue, they are compactly arranged, and along with this epidermis, this cuticle, that is the non-permeable water, non-permeable membrane is present, so it avoid the evaporation of water from inside the tissues. Okay, so this is about the epidermis. Now hypodermis. Just below the epidermis, there is a presence of hypodermis. Hypodermis means the cortex region, but in this case, the cortex is differentiated into two parts. That is, it is heterogeneous. The upper part of the cortex, which is just below the epidermis, which is present just below the epidermis, they are somewhat sclerenchymatous type. That is, sclerenchymatous tissues, they are present in this region. So multi-layered, that is 2, 3 or 4 to 5 layered, that is sclerenchymatous tissues, they are present just inside the epidermal layer. So this region of cortex, which contains the sclerenchymatous tissues, is known as the hypodermis. Hypo means below and dermis means epidermis, that is just below the epidermis there is presence of that is 3 to 4 layers or more than these, 3 layers of sclerenchymatous cells, and it is known as the hypodermis. Okay, the cells are as you know that the sclerenchymatous tissue, what type of they are thick walled and dead tissues. Okay, the cells are thick walled and compactly arranged. Okay, so the stem is present at the aerial region or the above the ground. So that stem facing the many hazardous situations, or they are facing the strong wind or herbivorous animals or all types of that is injuries they are facing there. So they have to develop this protective phenomena against that, to avoid or to protect themselves from that all these injurious things. Okay, so that's why these 3 to 4 layers sclerenchymatous layer is present just below the epidermis. So that part of the epidermal, that is that part of the outer membrane which are facing the outer side or the outer environment, is somewhat very much protective in nature, so they can resist the external injuries. Okay, so that's why epidermis with cuticle and also the hypodermis, they all together, they gives the mechanical strength and rigidity to that the stem. Hence they protect that stem, internal tissues of that stem from the external injuries. Okay, so this is the about the hypodermis, that is epidermis. After that hypodermis is present. Okay, then after hypodermis you can see in the picture, that is the colored patch is there after epidermis, and then after hypodermis there is a presence of blue colored cells, thin walled blue colored cells. They extend from the hypodermis up to the center of that stem. So this is the ground tissue. That ground tissue, they are present everywhere other than the hypodermis. Okay, so this ground tissue, that is the entire mass of parenchymatous cells next to the hypodermis, they forms a ground tissue. Okay, it is large, then multi-layered parenchymatous region, and it consists of rounded, or you can say somewhat oval or rounded parenchymatous cells with distinct intercellular spaces. They shows the intercellular because they are thin walled, and when they come together they form a space, that the space is formed automatically. So that's why they are as they are thin walled, they shows the intercellular spaces in between these. So they are large in number and they are multi-layered. Their cells, they are somewhat larger in size, and they are somewhat rounded or oval in shape. Okay, and it is not clearly differentiated into cortex, endodermis, pith, etc. As like in as we have seen in other that is anatomical structure of the dicotyledons and the monocotyledonous root and all these. So we can easily differentiate the cortex to the other from other parts of the plant body, but here there is no demarcation line of the endodermis, there is no presence of pith. That's why cortex is that is continuous up to the center. There is no any demarcation line in between this cortex and the still region. So this ground tissue is present everywhere, everywhere they spread. Okay, so this is the main characteristic of the maize stem plant. So you can easily identify that maize plant of seeing this type of structure. So these vascular bundles, they remain scattered in ground tissue. So they are not specific at the center, they are not specific under the endodermal or the pericycle region. But due to the absence of this pericycle, endodermis, and all these things in the maize stem, these vascular tissues or these vascular bundles, they remain scattered everywhere in the ground tissue. Okay, so the main function of it is to store what is, that is the ground tissue. The main function of ground tissue is to store water and food material. Okay, so this is about the ground tissue. So this ground tissue, which is made up of parenchymatous cells, Thin walled parenchymatous cells shows intercellular spaces, and they don't show any demarcation line, that is endodermis, and any pericycle is there. So these ground tissues, they spread everywhere, they scattered or they present everywhere from the hypodermis up to the center. The only the ground tissues are present, and on this ground tissue, in this ground tissue, in this ground tissue, there is a presence of vascular bundles, and also the as there is no demarcation line, these vascular bundles are also freely present or scattered everywhere in the ground tissue. Okay, so this is about the ground tissue. It is somewhat different from that of the previously that is studied all these anatomical characters. Okay, so this is the clue point, or this is the main point of this anatomy. Okay, then after ground tissue, we can see we will see the vascular bundles. So how the vascular bundles, they are present in this that is stem. So these vascular bundles, as they scattered all over the ground tissue, so they are many in number. Okay, so they are many in number and scattered in ground tissue. Okay, so each vascular bundle is somewhat oval in shape. As you can see in the picture, it is oval in shape and is conjoined, collateral, enlarged and closed. Okay, so this is as like that of the stem of the dicotyledon. It is also a conjoint and collateral. That means xylem and phloem tissue, they are present on the same radii. They are not present on the different radii as like that of the root, but they are present on the, that is xylem is above and phloem is below, or vice versa. Okay, but they are present on the same radii, that is on the same axis. Okay, so that's why they are conjoint collateral. But the main difference between the stem of dicotyledon and the stem of monocotyledon is that is they vascular bundles, they are closed type in case of monocotyledonous stem. Why it is closed? Because there is no presence of any cambium or the meristematic tissues, because there is no secondary growth in case of monocotyledonous stem. So the diameter of the stem, it will not increases in case of monocotyledonous stem. So you can easily see you can see that the in Mace stem, it is not that is enlarges in diameter, that is the trunk of that Mace stem is not enlarges, that is horizontally. It is lengthwise, it is increases, that is apical and the intercalary meristematic tissues, they are present, but lateral meristematic tissues, they are absent in the case of monocotyledonous stem. That's why secondary growth is not possible, and hence these lateral tissues, that meristematic tissues, they are present in the vascular tissues, that is in between the xylem and phloem. But in case of stem of monocot, it is absent, that cambium or the meristematic tissues, they are absent. Hence the vascular bundles, they are conjoint collateral but closed type. Okay, closed means there is no presence of meristematic tissues or cambium tissues. Okay, each vascular, now you can see in the picture that each vascular bundle, that is the xylem and phloem, you can see there, and this xylem and phloem is surrounded by some structure, that is this whole vascular bundle that is surrounded by a sheath of, that is covered by some type of cells, that is sclerenchymatous cells. So this vascular bundle is surrounded by a sheath of sclerenchymatous fibers called bundle sheath, which is specially developed on the two sides, that is upper side also and the lower side also. You can see in the picture that sclerenchymatous bundle sheath, that is labeled, is there, and again the upper part is also made up of this sclerenchymatous bundle sheath. So above and at the upper side and at the lower side of the this xylem and phloem tissues, there is a presence of covering, there is a presence of some protective covering, that is known as the bundle sheath, or the sclerenchymatous covering is also known as the bundle sheath, because it is covered the bundle, that is vascular bundle, hence it is known as bundle sheath. Sheath is like sheath means what? Which covers, sheath means which covers the any object. So sheath means covering, and the bundle sheath means this sheath covered the vascular bundle, hence it is known as bundle sheath, which is made up of sclerenchymatous protective tissue. Okay, because These vascular bundles, they do not have any demarcation or any endodermal or pericycle protective covering. Hence, they are free in ground tissue. So there is a there is a arrangement for the protection of these vascular bundles is the availability of this protective sheath. Okay, so these vascular bundles towards the there are two types of vascular bundles, and that means in on the basis of size there are two types. At the central region, that the when the you can see the vascular bundles, two vascular bundles at the peripheral side, or you can say at the hypodermal sides, and one of the vascular bundles towards the central side, or towards the center. They are located towards the center. So there is a difference in the size of these vascular bundles. So the vascular bundles which are present towards the peripheral sides, they are smaller in size than the vascular bundles they are present towards the central region. So this those vascular bundles which are facing towards the central region, they are larger in size, and those vascular bundles which are present towards the periphery, they are smaller in size. Okay, so there are these on the basis of size there are two types of vascular bundles. These vascular bundles, now the vascular tissues, that is xylem and phloem, how what type of tissues they are present, or what type of cells they are present in that vascular tissues, we will see in our next slide. That first of all we see the xylem. We focus only on xylem. This is the patch or the somewhat enlarged figure of the vascular bundle. It shows the sclerenchymatous sheath also, it shows phloem, it shows xylem tissues also. Okay, so only concentrate on the xylem tissues, that is the circular part or the large oval shape, that is circles they are present, or rounded shapes circles are present, that is known as the xylem part. Okay, so the xylem lies on inner side, that means the phloem is on the upper side and xylem is present under the phloem. That means these xylem tissues, they are facing towards the center of the stem, so they are lies in inner side. So it consists of protoxylem and metaxylem. Both types of vessels they are present, that is protoxylem and metaxylems. Then tracheids also seen, and xylem parenchyma is also present. Only the sclerenchyma is absent. So the xylem, it consists of four distinct vessels arranged in the form of letter Y. So you can see the two large vessels and two small vessels, and their arrangement is look like that of the it forms a Y-shaped structure. So the letter Y, you can write the letter Y, that is two arms and the one stalk. So this arrangement of vessels is also like that. So the two large vessels, they act as a two arms of the Y, and the two small protoxylem or the vessels, they act like the stalk of the Y. So it is looks like a letter, that is Y. Okay, so it consists of four distinct vessels and they are arranged in the form of letter Y, and a small number of tracheids arranged around them. So remaining part of these vessels, you can see the cells of this that portion, that other than the vessels, the some type of tissues they are present, that is known as the tracheal cells. So they are irregularly present in between these vessels. Okay, so the two metaxylem vessels are located at the upper two arms, that is two large vessels, they are facing towards the upward region, and two protoxylem, they are facing towards the central region. So the two metaxylem vessels, they are located at the upper two arms, and one or two protoxylem vessels at the base region. Okay, so two smaller vessels lying at the arm base constitute the protoxylem. So these small vessels, they constitute the protoxylem, and the larger vessels, they are known as the metaxylem. So in a mature bundle, the lowest protoxylem disintegrates and forms a cavity known as the that is xylem cavity. So this lowest region, lowest vessel, it get disintegrate, it destroyed. So that there is a cavity is formed in that area, that is the cavity of that xylem. Okay, cavity of that vascular bundle. Now let's see the phloem. Now these phloem parts, that is they are present upper to the that is these xylem tissues, that these metaxylem you can see in the pictures, the large vessels, circular shaped structure, and above that there is a presence of some type of large cells that is known as the phloem, and around that phloem there is a presence of bundle sheath. Okay, so coloring demarcation is there, you can easily see in this. So this phloem lies outer to the xylem. Okay, it consists of sieve tubes and companion cells. That means phloem is made up of four types of cells: sieve tubes, companion cells, phloem parenchyma, and phloem sclerenchyma. But in case of this mace or the monocotyledonous stem, the phloem is made up of only sieve tubes and companion cells. That means a phloem parenchyma and phloem sclerenchyma, or the fibers, they are absent. Then it can be distinguished into an outer crushed protophlloem and an inner metaphloem. So again there is a presence of two parts in phloem also, like that of the xylem, that the upper part is somewhat crushed, uh, crushed, that is you cannot see easily in the picture, but in some cases the that is the upper part of the phloem, that is the whole phloem upper, and it is divided into upper and lower. So the upper part, it is somewhat crushed type, so that crushed part is known as the protophlloem, and the remaining part, that is the inner part to that of the crushed protophlloem, it is the metaphlloem part. Okay, so again that is xylem also shows the metaxylem and protoxylem, and the phloem also show the protophlloem and the metaphloem. Okay, so this is about the vascular tissues. Okay, so let's have a quick look of the distinguishing characteristics of this diagnostic or the you can say the diagnostic features of monocotyledonous stem. So typical monocotyledonous stem, it shows the following characteristics that we have discussed from the right starting, that it shows that is stem is somewhat circular, that is the outer membrane or the outer epidermis is circular in shape, and it shows some depressions, like when you see the externally stem of the mace plant, it shows somewhat the depressed structures on the stem, because due to the presence of the lateral branches there, these lateral branches, the scars of that lateral branches, because these lateral branches arises from that area, there is somewhat depressed region is present on that part of this stem. So it is a it contains the epidermis, compactly arranged cells, epidermis, single layered cells, and thick cuticle is present, and but trichomes are absent. Okay, then uh, sorry, under to the that is epidermis, there is a presence of multi-layered sclerenchymatous hypodermis is present. Then ground tissue is not differentiated into cortex and pith. Okay, then hypodermis, as you can say hypodermis, only the cortex region, it is differentiated into hypodermis and ground tissue. Okay, so it has no distinct epidermis, sorry, endodermis and pericycle is present in these regions, and epidermis also contains the stomata and guard cells. Then the scattered vascular bundles, they are present, and which covers which shows the covering of sclerenchymatous bundle sheath. Okay, so these vascular bundles, they are oval, conjoined, collateral, and closed type. There is no distinction between pith and pith rays. Okay, and the xylem is enlarged. Phloem is represented only by the companion cells and the sieve tubes, and um, there is a presence of metaxylem and protoxylem. Again in phloem there is a presence of protophlloem and metaphloem. So this is just the overall quick look of the internal structure of the monocotyledonous mace stem. So this is how we can differentiate the dicotyledonous stem with that of the monocotyledonous stem. Yes. So in dicotyledonous stem of sunflower, we see the trichomes or multicellular hair-like projections from the epidermis. In the mace, it is not it is absent. Again, in case of dicotyledonous, we cannot see the hypodermis, but in case of monocotyledonous stem, it shows the hypodermal regions. So in case of dicotyledonous, the cortex is differentiated. It is differentiated because it contains the endodermis, pericycle. Okay, that is demarcation line. But in case of monocotyledonous stem, it is not distinguished. It does not show any endodermis or pericycle. Okay, so it is present all over the that is internally they are spread all over the body. Okay, in case of dicotyledonous stem, barrel shaped endodermis is present, then a sclerenchymatous pericycle is also present. It is absent in monocotyledonous stem. So in case of vascular tissues in dicotyledonous stem, they are conjoined, collateral and open type. But in case of monocotyledonous stem, they are conjoined, collateral and closed type. Okay, so both shows the enlarged condition, but in case of dicotyledonous stem, there is no protective covering around this vascular bundle, that is, you can say, the bundle sheath. But in case of monocotyledonous stem, it shows the vascular bundle shows the protective covering, that is known as the bundle sheath, which is made up of sclerenchymatous tissue. Okay, so this is the main differences in between the dicotyledonous stem and monocotyledonous stem. How you identified the internal that is structure of these two, by seeing the ground tissue and by seeing the vascular tissues and hypodermis. These are the three main points. On the basis of that, you can easily distinguish or differentiate the two types of stem. Okay, so this is about the stem of monocotyledonous plants, that is the maize plant. This is the end of the today's lecture, and the remaining part, that is the leaf part of the maize plant, we will see in our next lecture. Till now, uh, uh, again I remind you that um, up to this lecture, we have done we have completed, I think, 2 to 3 units up till now. So what about your experience? What about your feedback? Please give me your feedback, and whether you can understand or not. So what which that is a topic I have to revise, or I have to explain more in detail, or explain twice. So all these things, they are included in your feedback. About slides, a view of slides, about voice, about explanation, all these things you have to please convey me in your feedback. And you can personally give your feedback to me, or you can that is uh, give it in our group also. So better you can give me, you call me, or you text me your feedback, so that I will improve my lectures, or I will I will come to know which topic is I have to revise for you. Okay, so after completing this, I give you some type of assignments, I give you some type of test also. So please, before that, you please give your feedback, so about your understanding. So okay, that's over for today's lecture. We will see in our next lecture.